Hi, I'm Joy, and today I will talking about my video assignment BDY 3232 for the germination of common seed groups. And I'm from group 2, and my number matrix is S70249. So I will first talking about the background reading and literature review on crop species. Importance of seed germination in crop productions. So the seed germination is a complex process which a seed develops into a new plant and envelop various signals and is influenced by both intrinsic factors such as seed dormancy, seed size, seed viability, and extrinsic factors such as available food stores, wild water, oxygen, temperature, light, relative humidity, chemicals in the seed environment, and substrate. In the agricultural and ecological studies, germination experiments are commonly used to compare seed performance under controlled conditions and provide an understanding on specific germination strategies. Vegetable crop species such as chili and spinach are widely cultivated due to their nutritional value, short cycles, and economic importance. Study on germination characteristics help to improve cultivation and practices and contributes to sustainable food productions. So next is about the chili and also the spinach. So for the chili, its scientific name is about capsicum and is economically important fruit vegetable crops belonging to the family Solanaceae and is widely cultivated in America, Asia, Africa, and Mediterranean countries. Capsicum annum is perennial plant grown in climate-controlled greenhouse. Chile is a warm climate crop and requires 25 to until 27 degrees Celsius for optimal seed germination, and seed can be germinated around 3 to 9 days under suitable temperature conditions. So the seed is a disc-like with a deep chalazion depression and is covered by a parchment-like seed coat, which doesn't cause a mechanical restriction to germination. However, for the spinach, its scientific name is about Amaranthus spinosus. It's a fast-growing leafy vegetable widely cultivated as vegetable in tropical and subtropical regions. And the seeds for the spinach are shiny, black or brown, lenticular, and vary by size and also the species. And it can germinate based and in the warm soil and store well for short cycles. So for the first germination of spinach is within 2 to 4 days under optimal conditions like fluctuating temperatures of 30 slash 20 degrees Celsius or 35 slash 25 degrees Celsius with light exposure. So the next is about the methodology. So I will talk about the experiment design. And first of the objective, I want to compare the germination performance of the red chili and also the spinach. The germination experiment was conducted using two crop species and also two germination media. The first one is the filter paper medium, which is using the petri dish, and the second is the plotting soil medium, which using the polybag. So each species was tested with the three replicates, and during the experiment, there was no fertilizer was applied. So the crop species and germination medium will as the independent variables, while the times to first germination and germination percentage as dependent variables. So the left hand side, it shows that the filter paper medium of the petri dish and also the plot soil medium of the polybag. So I will start talking about the germination using the filter paper medium, which is the petri dish. So the first, I will place and fix the filter paper into the petri dish. Then place two different crops of the species seeds at consistent spacings, and a total of the three replicates for both crop species, such as the red chili with 3 seeds per replicate and spinach with 5 seeds per replicate. After that, moisten the filter paper and seeds with water. Then cover the petri dish and label with group number, crop species, and replicate number. So place the petri dish on the rack outside the laboratory and spray water daily or depends on the water condition of the seeds. After that, observe and record seed daily for first day seed germination, number of germinated seeds, and also the germination morphology. So the right hand side shows the uh, figure 1.1 to figure 1.3, which is a total of 3 packet of petri dish for the red chili. So each petri dish consists of 3 seeds for germination. For the below is for the spinach, it shows a total of 3 replicates of the petri dish for spinach, and each of the petri dish consists of 5 seeds for the germination. So next is the germination using potting soil medium, which is a poly bag. So the first is float the poly bag and fill with the soil, and then place 2 different crop of the species into the poly bag with 3 times replications, and a total of 8 seeds for the red chili are placed in each 8 poly bag. And for the spinach, the 10 seeds also placed in each of the 10 poly bag. Then water the soil to keep moisture, and then label each polybag with group number, cross species, and replicate number. And then place polybag on the rack outside the laboratory and spray water daily. And after that, observe and record seed daily for first day seed germination, number of germination seed, and also the seed morphology. So on the left hand side, it shows that a total of 3 replicates of polybag of the red chili and also spinach. So the total of 8 seeds for the red chili are placed in each 8 polybag, while spinach with a total of 10 seeds are placed in each 10 polybag for germination. The picture above show that the replicates of the polybag for red chili and also spinach with its replicate number. So for the red chili, is using the replicate number such as G2/R1, G2/R2, and also G2/R3. While, while for the spinach, using the G2/BR1 and also G2/B/R2 and also G2/B/R3. So for next, I will talking about the data collection and analysis. The observation and data was collected from uh, December 10th of 2025 until December of 25th, 2025. So the observation on December 16, 2025, it shows that the seed of the red chili in the petri dish consists of fungus for the G2R1, G2R2, and G2R3, while for the 12th of the 17th, 2025, the red chili in the petri dish, it shows the first germinate for G2R1 and also G2R2. In 10, in December of the 19th, 2025, and the red chili in the polybag, it shows the first germination for each of the polybags such as G2R1, G2R2, and also G2R3. However, the G2R2 have fungus on the seed for the petri dish. So the G2R3, it shows that it cannot germinate. Each seed consists of fungus, while for the germination percentage, you're using the formula below. So the left hand side shows that the first day for the data collection of the seed germinations. So the data for the both red chili and also spinach is started collected for the first day. For the right hand side, it is the first germination for the petri dish on 12/17, 2024, and it shows that the red chili for the petri dish was started germinated. For example, for the G2R1 and so the G2R2. So the next is on uh, December 19, 2025. It shows the first germination of the red chili in the polybag. The data shows that the red chili seeds for the polybag was germinated. A total of 1G2R2, 3G2R2, and also 2G2R3 of the red chili seed successfully germinated, while for the spinach seeds, there is no zero germination under the same conditions. For the right-hand side, it shows the paddy dish. The data shows that the red chili seeds for the paddy dish was germinated. A total of 2G2R2 and 3G2R2 of the red chili seeds successfully germinated. While for the below of the paddy dish, it shows that the spinach seeds, there was no seeds was successfully germinated. After that, after the 1 week of the first germination, for the paddy dish and also the poly bag, I will observe on December of the 25, 2025, and a total of 12 seeds of the red chili in the polybag successfully germinated, while the seeds of the spinach show the zero germination under some germination conditions. For the patty dish, a total of 7 red chili seeds G2R1, G2R2, and also G2R3 was successfully germinated, while the seeds of the spinach shows the zero germination under the same germination conditions. So after the data collection, I will talking about the result and also the discussions. The first germination of the red chili seeds was observed in the day 7 for the patty dish and day 9 for the polybag. For species, there was no germination was observed throughout the experimental period. So the below is the germination morphology for the red chili. It shows that there is apical morphology, while the spinach is undefined. So the right hand side of the picture shows that is the red chili seeds in the paddy. So after that is about the germination percentage. So the first is about the red chili. The first germination in the paddy dish shows 4. For the first germination polybag it shows 6. After 1 week of the first germination it shows that it increased until 12. So for the total seed in the paddy dish is about 9, and for the total seed polybag is 24. So after the calculation, it shows that the germination percentage for the paddy dish is 44.44 percentage, while for the red chili of the polybag, the germination percentage it shows that is about 25%. And for the spinach, the first germination and also the for the paddy dish and also polybag is shows the 0, and after 1 week also still in the same situation which is at the 0 number for the total seed for the patty dish which is 15, and also the total seed for the body bag is 30. So after the calculation, it shows that the germination percentage for the patty dish also the body bag is shows a 0 number of the percentage. The result shows that chili germinates earlier than spinach. This difference can be explained by seed characteristic and physiological response. Chili seeds are larger and can absorb more water consistently compared to spinach seeds, which are very small and sensitive to water, environment, and also moisture availability. The experiment was controlled by maintaining consistent environmental conditions and water frequencies. However, there are some limitations including outdoor temperature, affecting moisture levels, potential variation in seed quality, which can influence the germination time of seeds. Although these limitations caused the germination of seeds, the morphology was designed to ensure consistency across treatments while allowing comparison between species and germination medium. So, in the conclusion, the chili seeds and spinach seeds show two different cross species with different germination behaviors. The chili seeds germinated earlier than spinach seeds under same germination conditions, despite the literature review shows that spinach seeds is germinated faster than chili seed. It also shows that the germination is influenced by surrounding environmental conditions and seed characteristics such as seed quality and moisture conditions. Filter paper in package of seeds germinate faster than polybag of seeds due to more consistent moisture conditions. Besides that, the occurrence of fungus in surrounding of seed can lead to unsuccessful seed germination. This can disturb the seed condition and germination. So last but not least, this is my reference during my study for the germination of the seeds. And also, thank you for the listening.